Hello everybody. Today I'm going to talk about cell division, like mitosis and meiosis. This is lesson 1 presented by Sally Ahmed Khalil. So first I would like to tell you what is cell cycle. The cell cycle is a process where there is two phases. We have interphase and mitosis or meiosis happens. So here in the interphase we recognize we have G1, S phase and G2 phase. In the G1 we recognize that the cells start to grow, and here in S phase they start to produce DNA and multiplicate the DNA, and here in G2 the cell continue to grow and they produce extra protein. Here we will recognize the green part where we contain the process of mitosis or meiosis happens. So we could obtain the prophase. We have early and late prophase, and metaphase, anaphase, telophase, and at the end cytokinesis, where the cell divided to here in the process of cytokinesis. So I will talk about them later, those steps. And here is the site of this picture. So I would really like to talk first about the difference between mitosis and meiosis. So here mitosis, we recognize it is specific for the production of somatic cells, like the cells which are in your skin or your tissue, any organs in your body. So all of these organs are produced by mitosis. So mitosis, at the end of mitosis, you could obtain two cells which are identical in their genetic materials, and they are diploid. They contain 46 chromosomes. That means each cell contains 2N. Okay, but here on the other hand, meiosis, we could recognize that it is happening within the reproductive cells to produce sperms and eggs. It gives you 4 cells at the end of the reproduction, and you could recognize that the cells are different from each other. They contain different genetic materials and they are haploid. That means they contain 23 chromosomes each one. So here if you have 4 sperms, okay, formed, those cells are 4 sperms in a male. We recognize in the female she will form 1 egg and 3 polar bodies. The polar bodies are not important at all, but the egg is important for fertilization. When it is fertilized, the egg now it contains n number of chromosomes, and the sperms contain n number of chromosomes, so they will form the zygote, which contains 2N or 46 chromosomes, and this zygote will form later the baby. So now I will talk about mitosis in details. We recognize here the prophase. Here the chromosome is condensed here, and the nuclear membrane will disappear in this phase, and the centrioles here are going apart from each other. In metaphase, we recognize that the chromosomes are arranged within the equator of the cell, and the spindle fibers are attached to the centromere of the chromosome. The centromere is the part which attach both sides of the chromosome together here. Okay, and anaphase, here the chromosomes, the sister chromosomes are going apart. They are separated here from each other, and they are going apart toward the pole of the cell. The spindle fibers here are pulling them. Okay, and telophase, here we recognize that the chromosomes here are they arrive to the pole of the cell, and the cytoplasm here will begin to divide. And here we cytokinesis, where the process of separating the cells occur. Okay, in this process of cytokinesis. Now I will give you a hint about the difference between animal cell and plant cell. As you know, plant cells contain cell wall, however animal cells don't. Okay, so but here in animal cell they have centrioles, okay, which produce muscle spindle fibers, but here in plant cells they don't have centrioles, but they could produce the spindle fibers. All the other processes like metaphase, all the other phases like metaphase, anaphase, they are identical, except here for telophase we recognize when the cells divide here they produce a cell wall between the two cells, but here in the cytokinesis, to separate the two cells, they separate it by pinching them into two. So here I will talk about crossover before talking about meiosis because this is very important in this process of division. So here, if you remember, I was talking about interphase before, where I told you that the cell is preparing itself into to divide. So it prepares itself through passing to the G1, S, and G2 phase, and then it enters to process of meiosis and mitosis. But here we have early prophase 1 and late prophase 1. In early prophase 1, we have homologous pair of chromosomes. They are arranged beside each other, okay? But there is no crossover happening. What is homologous chromosome? Homologous chromosomes, they are chromosomes which are identical in their size, shape, and for example, the centromere is located here in the middle. This is another chromosome. The centromere is located in the border here, okay? So those are homologous chromosomes. They are the same in length. So they are attached near each other, coming near each other in the prophase 1. They exchange their genetic materials, as you see here. They are crossing over here. So after that, we will get a mixed chromosome with different genetic materials. And the spindle fibers here are responsible for pulling them apart. Okay. So here we will see in meiosis, we have prophase 1, where the chromosomes here are arranged, are condensed, and they are crossing over. So now they are exchanging their genetic materials. This is the most important part in the meiosis, okay? Because this is not happening in the mitosis. And this is the reason why you are different from your father or your brother in your genetic or inherited materials. From this through this part, okay, through prophase 1. And here metaphase 1, we recognize that homologous pairs of chromosomes are aligned here in the equator of the cell, and the spindle fibers, as you see, are attached to the centromere of the chromosome. And in anaphase 1, they are the homologous pair of chromosomes, they are going apart here, and the spindle fibers are pulling them apart towards the cell, the pole of the cell. And telophase 1, here we could recognize that cytoplasm begins to divide. And here in cytokinesis, it gives these two cells. Here, the prophase 2, the same happening, condensation of the chromosomes, the identical chromosome here. And here metaphase, the series are arranged. These chromosomes are arranged within the equator of the cell, and spindle fibers are attached to the centromere, the same happening. Okay, anaphase sister here, we could recognize the sister chromatid are separating apart from each other to go to the pole of the cell. After that, we could recognize the cytoplasm is start to separate in the telophase 2. And here the cytokinesis, it produces those 4 cells with different genetic materials. So this is meiosis. Okay, in brief, we could recognize we have here prophase 1, metaphase 2, here condensation of protein, here alignment of DNA within the equator of the cell, here anaphase 1, where you could recognize the sister chromosomes are apart from each other. Here telophase 1, the cytoplasm start to divide, and the same prophase 2, here condensation, here alignment within the pole, the center of the cell. Here there is separating apart in anaphase, and in telophase 2, here the cells are divided to give 4 cells. They are not identical in their genetic materials, and after that cytokinesis happens, and after that it enters into each cell, enters again in interphase, which contains the G1, S, and G2. So now I will give you a summary of what I mentioned. Okay, here in mitosis, we recognize we have prophase, metaphase, prophase, metaphase, anaphase, telophase, and this is cytokinesis, where the cell divide again to give 2 cells, but they are identical in genetic materials. They are exactly the same as their parent cell. But here in meiosis, we recognize that we have prophase 1, where the crossover happens and there is exchange in the genetic materials. Here metaphase 1, where they are arranged here, and spindle fibers are attached to the centromere here of each chromosome. And a phase 1, where the homologous chromosome are going apart here to the pole of the cell. Telophase 1, where the cytoplasm start here to divide, and here by cytokinesis they give those two cells, but they have two genetic materials which are different from the original cell. Okay, and here after that we will go to the same prophase 2. Here condensation happens. Metaphase 2, where there is alignment here, is in the cell, the center of the cell. Anaphase 2, they are separated here. The sister chromosomes are separated apart from each other. And telophase 2, here the cell start to divide through the cytoplasm. And here it gives you 4 cells, but they are not identical in their genetic materials. Here it gives you 2 cells. Here it gives you 4 cells. Okay, here they are identical in genetic materials. Here they are not identical in their genetic materials. So here for communication, I just want you to know that you could ask questions or leave any comment, or I would like also and appreciate if you read this lesson. And here I would also like you to know that those are the site of communication. You could communicate through a Twitter account, or through YouTube, or through WordPress. Okay, or leaving any comment. Here are the references that I used through this presentation. And I would like here to give you and ask you some questions. And if you would like to ask me some questions, I would really appreciate that. So I will ask you some questions, including true or false, if in the space, or any other questions, and I will give you a feedback of your answer if you would like. And you could send a comment, and we could contact to either as a sort of contact through the sites that I mentioned before. So I will do the best to help you. I will try to do my best. So here are the questions that I ask you for this lesson, and I would really appreciate if you give me a feedback on this lesson. Thank you very much. And I hope that you enjoyed this lesson, and have a great day.